there. The following video is meant for those that are taking microbiology class, especially the general microbiology, and one of the first labs and techniques that we need to learn and understand is how to transfer bacteria, yeast, or any microorganisms, could be mold, from plates to different other media. It could be another plate, it could be tubes, broth, or tubes that have agar and are slanted. Now, things that you should know by now is that this is a petri dish. How we make the petri dish? Well, this is just plastic, right? And in here we put nutrients and a substance that we call agar. The agar is the agent that solidifies those nutrients, that broth, that liquid that has nutrients, and we get something like this. Textures, you can see it on the screen, is pretty much like jello. So in this case, I want you to see the actual plate and how the agar look like as a close-up. You can see that the agar, it's breakable, very easy to break. Even when you're actually touching it with the inoculation loop, it's actually easy to break or to penetrate. But at the same time, if you do it smoothly on the top, you don't break the actual agar. So just wanted you to take a look at that. These plates are a little bit old, they're a little bit dehydrated. Sometimes it's a little bit thicker than that. But I mean, they're fine. Just want you to at least see that.
There are two ways to get those agar plates. Either you buy it already done and solid, sterile, or you can buy the empty petri dishes, as you can see on your right side of the screen, and then you pretty much have to make your agar and pour it on those plates. So this is the way that you do it. First, you need to autoclave your nutrient agar. If you're actually using nutrient agar, as you can see here, it can be whatever type of media. And if you're doing plates, of course, I have to have nutrients and, of course, the solidified agent, which is the agar. The next thing that you need are the plates. This should be sterile plates because you're adding nutrient agar to it. In this case, I'm doing tubes, slanted tubes, and of course you need a Bunsen burner or any burner that you would have. In this case is an ethanol burner. So the way that it goes is, as you can see here, I open that media shake flask. In this case, this is hot right now, should be around 45 to 50, 55 degrees. Remember, the agar would get solid around 40 degrees. So you don't want to wait until it's 40 degrees because then you would have solid agar in your shake flask. So I stack the petri dishes and then I pour 15 to 20 mils in there. Now, there's no way that I can put 20 mils exactly in every plate, but experience would give you the edge. Of course, it was very hot, so you should be using your specific tools for this. So again, I'd stack them in like 3 or 4. And every time that you pour it, you must go through the flames. Now for the slanted tubes, the same principles applies. In this case, these are sterile plastic tubes, so you don't have to flame those. So you just flame the Ellermager or shake flask, pour 5 to 6 mils in there, always flame the shake flask. After you pour that, then you close the tubes, and as you see there, I inclined those, that way it form that slanted portion, and when it gets solid, you will have a slanted tubes. Did the same thing here, so you can see over there that they are slanted, so you create that surface in that tube that you can pretty much inoculate your bacteria in there. Now, I always get the question, why would you do something in plates versus tubes? Remember, in plates we use 20 to 25 mils of agar, and there's like a big space that we can do streak plates, or we can have multiple bacteria in there to pick. Normally when we use tubes are just for pure cultures, since we know what specific bacteria or microorganism we have in that specific tube. So we use it for pure culture because it's a small area for the bacteria or microorganism to grow, and at the same time we use less materials. We would use 5 to 6 mils per tube.
So approximately we put in this type of plates, let's say 15 to 20 mils of that substance in there. That way we can actually grow the microorganism on top, and then we can actually observe them. So these techniques are from those that are moving from one specific media, let's say that we imagine that we have bacteria in here, to move it into this plate. So you can actually see how the transfers are done. One of the most important things is to have a Bunsen burner. Okay, so if we don't have a biosafety cabinet, which pretty much is a clean environment, that probably some of them have either HEPA filters like the BSE biosafety cabinet, or a laminar flow booth, which pretty much is to push the air from one source to the other. So it's either from here, from one side to the other, or laminar flow can be from top to the bottom, or from the back to the front. I'm going to show you my homemade laminar flow booth in a second. So you can see in here that you can actually see the HEPA filter, and the flow goes from the back to the front. So that way, if you imagine we have here this environment from there to here, so all the air, the clean air is coming from there to here, so nothing is happening in here. I can actually open a plate like that and then won't get any contamination. Imagine that you have spores hanging around in the air, right? It will not go there because there's a flow of clean air that has been cleaned 99.9%, right? No bacteria, no spores, etc., and has been cleaned, so all the air would come this way. All right, so that's one thing. So if we don't have that, then we need a Bunsen burner. In this case, it's not a Bunsen burner. It's pretty much a burner that has ethanol. So, but you need fire. Why fire? Because we need to sterilize your inoculation loop. When you sterilize your inoculation loop, you need to put it, inoculation loop, exactly on the flame, and you will see when it's pretty much literally red hot. After it's literally red hot, then you can continue doing your transfers. I have a couple of examples that I'm going to show you later on. But for example, let's say that we're transferring from a tube. Very important that with the pinky, normally you take the cap, right? Now, this tube is plastic, so I cannot flame it too much because then I'm going to melt it. Normally these tubes in the lab, we use glass. So pretty much you take it with your pinky, right? The theory is you take it with your pinky, you always try to work close to the flame. You flame it 2 or 3 times, then when you finish doing what you're doing, you cap it again, and this goes to the rack. You cannot just leave your tube in there because it could be dangerous, because if you have a bacteria here, you can have a spill. So you always have to put it in the rack. All right, so let's see how it is. Imagine this is a culture, and why I'm calling it a culture versus a broth. So you should know by now that a culture is when you have a broth, a tube with broth, or nutrient broth, can be whatever that they have nutrients, and the actual microorganism is growing in here. That's why we call it a culture. If it's sterile, like this one, there's nothing growing in there, it's just called a media, right? Media tube with just medium, okay? Now, let's imagine that this is a culture. So the way that you do it is close to your flame. Flame your loop. When it's red hot, with your pinky, remove the cap. With your other hand, flame it 2 or 3 times. Then with your inoculation loop, right, leave it close to the flame, not very close that you can get burned. Then you take your sample. After you take your sample, don't go like that. Why? Because now it's not sterile. It does have the bacteria, but you're contaminating with possible contaminants that we have here. So you always put it inside and leave it close to the flame. Not close enough that you can burn it, but let's say like 4 to 6 inches from there. Because this area is actually warm. If you put your hand there, you feel that it's warm. So after that, with your pinky, right, the cap that you have in your pinky, you close the culture, put it in your rack. Then now you're ready to either transfer into a tube or to a plate. If you're transferred to a tube, let's say that this is a sterile tube with medium, right? Then the same thing. You have the bacteria in there. You have to do this quick. So you open it with your pinky, flame 2 or 3 times. Your bacteria is here. Mix it in there because you want the bacteria to be mixed in that broth. After that, you flame, cap it. And after you cap it, rack. And after that, you flame your loop. Right? It is the same thing if you're going, let's say, from a broth or slant to a broth or slant. It's the same thing. Now, when you're going to the broth, you pretty much just mix it inside the broth, right? Remove the bacteria and mix it inside the broth. When you're going from, let's say, a slant, then you take a small portion of your bacteria, and if you're transferring to a broth, pretty much you just shake it. If you're transferring to a slant, or Agar slanted, a tube that is slanted, then you can do like a streak on the top. Okay? If you're going from any media to the plate, that's a different story, and I'm going to show you in the following video. So you always have to sterilize your loop, always. So as soon as your loop is hot, then of course you can continue. So how do you know it's hot? 2 things. You can put it here and then you literally wait until it's red hot. Another technique, a good technique, would be you get your fingers and touch it, and if it burns, then it's hot. No, please don't do that. That's a dad joke in there. All right, so after it's pretty much sterile, then you try to get that loop a little bit cold, right? Because right now it's like literally red hot. But you leave it closer to the fire. Why? Because this is very hot in here. So there's nothing really, no bacteria, spores that are get here. Now if there's like some spore hanging around, it would get burned in there. And bacteria, they don't have, unless they are on saliva or drops of water, they don't have wings to fly away. Okay, so pretty much what you do is you have to, with this finger, remove the cap, hold it in there, right? Flame the tube. Now these tubes are plastic, so I shouldn't be flaming this, but again, because it's for the example, flame it. After that, you take your bacteria. If you hear a something like that, then you're burning your broth. All right, so you take it, now it's in there, put your cap, which you have it in there in your finger. After you seal it, you put it on a rack, always in a rack. Now you have the bacteria in there, then you come, you open the plate, 45 degrees, just like that. If you open it like that, what's gonna happen? You're exposing the plate, especially if you're talking, like me right now. So you don't want to do that. You open it 45 degrees and then you do your streak, just like that. Now, since I want you to see something on a streak plate of isolation on this one, I'm not going to use this one, and I'm going to do a regular streak in this one. So when you do that, then you pretty much just open it 45 degrees and then just perform your streak in there, just like that. So you can see marks in there, right? And then that would mark the surface of it. Why I don't want to do that in this one? Because I really want for you to see the streak plate of isolation using this plate. Because you can actually see the marks now, although you're not going to see the bacteria right now, but at least you can see the marks. So imagine that we still have the bacteria in there. So you come here and then you do your inoculation in the first quadrant. After that, you close it. Then you need to sterilize your inoculation loop. And after is sterile, then you're done with the first quadrant. Now for the second quadrant, now let me just show you. I'm going to open it just for the example. So if you can see there the streaks. There's nothing in here, nothing in here, nothing here. Remember, we're going to have 4 quadrants, imaginary quadrants. So this is the first one, right? So there's no marks in here. Okay, let's close it. Now, would you go back to your culture? No. Why? Because you already inoculated that in there. And remember, the streak plate of isolation, what we want to do in a streak plate of isolation is from here to move it, right? To dilute this sample. If we use number, remember we have, let's say, 100 microorganisms spread in these areas that we streaked. If I come now and touch this area from, let's say, 2 to 5 times, 2 to 6 times, we are only taking or moving from this quadrant to the second one a few bacteria from, let's say, that imaginary number 100, right? So let's say that we move 50 or 25, okay? So now we have 100, 25 in the second quadrant, and then we dilute it again. Maybe from 25 we have 10, and then from 10 we have 5, and at the end we have isolated colonies, okay? So now you need to make sure that you know what you're doing in this case, because you can see the marks that are in there. So this is the quadrant in there. So normally we do that. We finish this quadrant, we close it, then we sterilize. At that point, you move your plate. You move your plate only 90 degrees. Why? Because now you're doing the second quadrant. And then you want to touch this specific area, which there's bacteria already in there, although you don't see it because we haven't incubated yet. So you come here, right? And you come 1, 2, 3, 4, 5, and then you spread. Then you spread that bacteria in here. And don't forget to sterilize. No, we're not spreading the bacteria. So now you have, let me open it that way you can see the streak. So you can see it in here, and now you have it in there. You can actually count the times that I touch in there. The times that I actually touch this area here. So now again, close it, of course. Sterilize, then you come here to the third quadrant. And another thing that I want you to hear, let me see if I can actually you can actually hear is this. When it's red hot, there's no bacteria in here. One technique that some people use, sometimes I use it, is like if you really want to hurry up, then you pretty much touch the agar. Hopefully you hear it. You touch the agar in an area that there's nothing because you don't want to kill anything. And then you start to do the streak plate. 1, 2, 3, 4, 5. And then you spread and sterilize, and you do the same thing on the last quadrant. Okay? And you do the same thing. Okay? And that's it. Sterilize this. Put it aside, and now I'm just gonna close this. All right? So you can actually see, let me try to look for the light. You can actually see the marks in there. Especially I want you to see the last quadrant. So in this case, you can see the first one in here, second one in there, third one, and the last quadrant, you can see it in there. That we go from here, from this one, all the way to here, and we have this streak plate. I'm going to show you a plate that you can see the different colonies spread out, and this is pretty much what we want. That all those colonies that maybe were here combined, maybe there were 2-3 different bacteria, at the end we can isolate them.
Let's do a streak plate of isolation on a faster speed. So I'm flaming, picking up the bacteria, inoculating that, streaking on the first quadrant. After that, flaming. You see how I turn the plate, second quadrant, streaked, touches at least 4 times, first quadrant, again, flaming, waiting for that to get warm instead of being hot. That's the third quadrant in there, flame again, and then just wait and perform the fourth quadrant. And there you have it.